क्या फोटो
निकाले
और मैं डेट टाइम पर विजिट करेंगे डेट टाइम रिसर्चर्स हमको भी स्क्रीनशॉट निकालना है क्या करता रहता है ये हर दिन नए नए बंदे ज्वाइन होते हैं इसमें रिसर्चर्स एंड
पढ़ पा रहे क्या लिखा है? ये एंटी जीएसटी के नीचे क्या लिखे हैं, कुछ पढ़ पा रहे क्या?
सिग्मा
एंटी जीएसटी नीचे क्या लिखे हैं?
सिग्मा कुछ लिखा है
सिग्मा लिखे हैं क्या?
सिग्मा तो लिखा है सिग्मा क्यू क्वाड्री लिखना चाहा ऐसा कुछ नहीं है सिग्मा है नीचे hello hopefully I am audible and the screen is visible. We'll be starting in 1 or 2 minutes. So good afternoon everyone, and welcome to for the week 6 live session for the course Introduction to Proteogenomics. So today we'll start this session. So for this week, so you have gone through the lecture that different workflows are there, which and different available tools and the software with which you can try to identify the different novel peptides, or the try to integrate the genomics and the proteomics data and find the proteogenomics, try to analyze those data and to correlate those data for the RNA-seq data or the whole exome data or the proteomics data. So I'd like to go through this paper which has provided different tools and software. Different tools and software with which you can know that which tool to use for to which step, since you know that there are many steps required to analyze the data fully and to interpret those data, and many of which tools are also been assembled in the Galaxy platform, as shown in in this week's lecture. So here are the different steps for the analysis of the whole exome sequencing data, to identify SNVs or the RNA-seq data, to identify, to construct those transcripts, and then finally adding those peptides raw sequences, and to identify the novel peptides and the transcripts. So if you go through this paper, you will be knowing the different steps, and also for the different novel peptides, or which different application can be done, different applications of those particular mutated or novel peptides for a particular patient or a particular sample. So you can go through it and know its different applications as well. So here, if you go to this paper and try to look at the table, so they will be knowing about the different tools, and when which step you can perform those tools, you can use those tools to analyze your data. So you can just download it. So you can see the computational tool. So the first is the whole exome sequencing analysis. For this first tool is that one tools for alignment. So you need a first QC tool. So what is the description, where is the link, the references, as well as why, at which step, and what does the particular tool does. So you can go through it and you can identify and you can get idea about all the tools, most popular tools, mostly available tools that are being used for the analysis of this type of data, and how they does, what is the input file for this, like align the first Q file, and the output is in the SAM format. For the when you align the align it, generally it gets in the SAM format, but also you can interchange the formats, like it also gets in the binary alignment map file, but that you cannot visualize, so you can convert it into the different formatters are there, where you can convert the different formats, different types of alignment tools, to tools for single nucleotide variants calling, at which tool is used for what purposes, for the structural variance identifications, as well as for the VCF annotation, as well as for variant filtration from databases. So different databases are like there, like 1000 genomes, TBSNP, Cosmic, where you can get an idea of the different reported somatic mutations or the single nucleotide polymorphisms that have been reported by different researchers, and how you can incorporate, or how you can integrate with your data, like Cosmic, then genome aggregation database, then tools for determining significant somatic mutations. So there are different kinds of mutations you will get. So which are were the significant ones? So identifying, then for estimating copy number alterations, CNV6, SEC. So among these tools also you can go and search in the Galaxy web server that if they are present or not, and you can try to integrate into your workflow. Exome CNV, VARSCAN2, so tools for different pathways analysis of the different proteins you identified. Then there are some of the tools that have integrated many tools and created one pipeline, like FastQ to VCF. So this software will take your input FastQ file and generally give your output as a VCF file. Then like Impact, which are integrates, just you have to give the input file and it will give automatically generate your output file. Then tools for prediction of impacts. Okay, so SNPF, STM, Shift. So these are different. Some are online tool. So here you can go, if you go and go through it, so you can find out that it is written that some are online tool, which are present, some are used to be downloaded and some to be used. So you also can search in the Galaxy web servers and different online platforms that are present. So you can get an idea and can use any of these tools, and also there are some specialized tools and software that have been implemented in various studies, like the GATK pipeline, or some other pipeline established by some of the groups. So you can search for those specific tools and can also do the analysis. So these are so another is that differentiation of the, when you identify the mutations, so how can you differentiate between passenger and driver mutations. Then transcriptomics, RNA-seq analysis that has been shown in the videos. So HTQC, fast text toolkit. So with different types of aligners. Then for RNA sequence specific quality check. Then for transcriptome construction, then you need to follow the step of differential expression analysis to find out the gene counts that are differentiating between your two conditions, and then combining whole exome sequence and RNA sequence data to find the variants. And these are the different tools, and then finally forming the customized database. So this you all can go through it to know about the different tools and databases. So now moving on. So now you can all have gone through the Galaxy web server. So let us look at that how, since Galaxy web server hands-on will be difficult because it will generally takes a long data server, takes time to be analyzed, and also there remains a limit. And also you can, for instance, here you can log in and register. So after your registration, so you can see that I had already has some data in the history. So this is the FastQ file. Okay, so if you can look at it, so this is the FastQ file. So if you go to the SRA data, right, SRA NCBI. So if you go and search SRR 101 47633. So you can see the study. What is the study? Okay, so this is a whole exome sequence, and then genomic DNA was extracted using what kits, what platform was used, like Next 550 Lumina sequencer was used, and it is of 125 base pair paired end. So it is done using paired end sequencing, and this different samples, and the strategy is whole exome sequence and paired, and the instrument is Nextech 550, and the size is of the data is 4.9 GB. So after retrieving from SRA toolkit, if you remember that I've asked that said that you can retrieve the data set in FASTQ format from SRA using SRA toolkit. So after retrieving your data from SRA toolkit, so you can upload it in Galaxy. Better will be uh, using FTP service, so that it takes less time to upload it. So since it is paired end sequencer has been done for this data, so you can see that here 1 and 2, so the two reads are been uploaded here. You can easily see that after uploading. So now the first Q files are ready. So the first step is to retrieve the data, and if you are generating your own data, so then that is fine and good, and you can continue with your data, or otherwise you can retrieve the data from different data of other researchers and can look at how it is there. So you can see that it is the sequences are here, and after the uploading is done, so we generally do is that we go and look for the fast QC report to check the quality check of the report. So in this history, in this workflow, so after we have uploaded it, so we are looking at the how much is the what is the quality of the data, genomics data. So using in this here, if you search fast QC, so you can go and read quality reports. So you can go and give that in SRR data 1 or SRR data 2, and you can just execute it. Okay, so you can see that new workflow has been generated where you can execute the fast QC report. So here if you check the report, so if you click on this icon, you can go and check at the report. So here it is the fast QC report. So it is a basic statistics. So you can see the file name that you have given will come, and so it will give the percentage of the GC sequences, sequence length. So you know that it is of 125 base pair, and it is showing the same. Then per base sequence quality. So the thing is that generally the sequences, when this NGS is done, so generally it is done on the basis of the probability of the base calling. So if the scores are above Q30, that means that the sequence that has been called at that particular position is more than 90%, means is accurate, and it is not by random or by chance, and it is that for the particular position only. So we take the samples which are having more than Q30 quality score. Generally more Q30 to Q40 in the range those samples are there, sequences are there, then only we take forward for further analysis. Per sequence quality scores, so you will get this full report. So the GC content percentage, sequence length distribution, so you can see also that everything has been passed. Also you will get the adapter content, that percentage of adapters, sequence duplication level, sequence length distribution, and all. So you will get over full statistics here. So you can from here go and download the files also, the report, if you want to save it in your local drive, so you can in your local PC. So you can just go and download this drives, download this data. So you can see that it takes time, and it will show the data. So So in this way, this is the first step or the first QC file. So if we go here and look at for the different tools that have been mentioned here, correct. So after the first QC, so if you have multiple first QC files, you can combine with multi QC. So now if you search PCAT tools here, so here it is not there, so you can look for trim gallows. So trim gallows also works on the same thing, that it will trim the adapter sequences, so which are there in the Galaxy server. You can try to identify and follow and okay. So filter base to cube, last base to cube cuts base sequences in a first cube file, so it will trim the sequences, but we need the trim gallows. So here you can check the data. So whichever sequences you want, whichever tools you want, you can try to find out and can function on those data. So it will automatically take the input file and perform the output file, and in this way will create a workflow, and you can save the workflows in this in your Galaxy web server. So here it is showing that the data set is large and it is showing only the first megabyte. So if you can say that okay, I will want to show the full length, so it has done nothing but the adapter sequences that are used during the sequencing steps. So it has streamed those sequences. So this is for the uh, trim gallery. Also you can use card data as well. So if you can say that PR dent, so you will say that okay, so you will give first q file, so one dot first q and 2 dot first q. So and you will give that yes, you want the 3' end adapters as well as front end adapters. Okay, so if you want to cut adapter before the adapter trimming, if you want to cut some bases or not, so by this, so you can select that what are the output files you want, that the statistics, and also you can use this file also with multi QC. Okay, and then you can execute it. So actually it takes little bit time to for this job, because the data size is huge for this kind of big data, but using easily the all those tools have been integrated in Galaxy web server, and you can easily download one of your data set, retrieve those data set, and can start working on building up your own workflow, customized workflow for a specific set of information you want to retrieve from your data. So like this it will start. So like see, you can see that fast QC data, as it has also been done, and it has given you the report. Similarly the cut will started doing, and it will perform the results. So next, after this, if you go here and you can check that you want to align it. So if you have bit of bow tie too, if you go and search, so you have, right. So you can say again PR dent, so if you need, so it wants in the data set as the data type in FASTA, so you can convert the data formats. uh, so using this, first few files only. So you can input these first few files and it will give you the FASTA file. So from here you can also change the different formats, and then finally you can go ahead with alignment. So you can check that these tools are already available, so you can go ahead and look for this tool. For example, SAM tools are one of the important tools because it helps in converting. Okay, so it also gives the quality check for your alignment tool. The SAM tool reports stats of the BAM index file. So when you align it, you will get a BAM file. So you cannot visualize those files. For visualizing in IGV, you want to convert it into SAM file, that is a sequence alignment map. So, and also if you want for the different statistics of what are the different statistics of how the alignment quality is done and there, so you will get through this software. Still it is cut adapted is only running, so you can find out these different tools. Now if you go for the RNA-seq data, so if you want to align with star, okay, so if you so there are so here I have given in the PPT the different quality control like trim galore, multi QC, okay, compute quality statistics, SAM, BAM, changing, and for the RNA-seq also there are different tools. So if you search here only, okay, so here you can see that transcript integrity number, RNA integrity, and then read GC, GPF, compare, HTC count, star, RNA star, which helps you to map like bow tie, and this is done for this is done for the whole exome sequence or the genomic sequences. For the RNA sequence faster fast Q file, you can uh, for the RNA star you can, then the transcriptome construction you can create using different tools. Then you can finally using AJAR you can find the differentially expressed RNA, which you can compare with your proteomics data. So here AJR will perform differential expression of count data. String tie will merge the different transcripts with the GTF files, so the gene transcript files. So you can go ahead and search with all these tools in this software, and also can go through the paper and can look at the different tools that are used for different analysis, and you can create your customized workflow, and with the different data set you can identify. So hopefully it is clear till now, right? So once it is done with the cut adapt, we can look at the scores, that what was there before filtering, before the adapters, how the sequence was looking with the adapters and all, and now after the cut adapt, that which will cut all the adapters sequences after it is trimmed, how the data is looking and how the sequence quality has improved or not. So you can see that the after alignment, so using this same sequence, if you visualize in IGV, you can find out chromosome wise that how it has been aligned and where there are the differences with the genomic sequences. Are there any differences or anything, or the single variants are there or not? So you can go and look at it. So this is the example, and also what I told about SAM tools IDIC stats, where you want to have a alignment quality check. So after it is aligned, how much is the quality check or what are the statistics? So you can use SAM tools IDIC stats. It will give a report where you will say the chromosome wise report, that how much, so you can see that the column 1 will be the reference sequence identifier when the sequence has been identified as chromosome 10. Number 2 is the reference sequence length, what is the sequence length. Third is the number of mapped reads and number of placed and but unmapped reads, so which are present but could not be mapped. So in this way you can get an idea about how much is your alignment quality is there, or you need any further improvement, or it is fine to go ahead with. So you can understand through different statistical analysis software for the alignment. So now another thing is that since we were discussing about different tools for the integration of the proteogenomics approach, so we till previous week we had came to about annotating the variants using ANOVA. So now is that we require a customized, after annotating the variants, we need to have a customized protein database from which the protein data or the mass spectrometric data will be identified, will be searched through to identify the novel peptide or the novel protein or the mutated protein sequences in your data from the genome exome or the RNA sequencing that you have performed. So for this, there is a tool called the custom prot bj, so which is used for the customized protein database construction. So it can build, so if you go to it and search, and you will find that it can build a customized database based on single VCF file, build a customized database based on multiple VCF files from a sample, and also you can build a customized database based on multiple VCF files from multiple samples. So depending on your application or what you want to check or what you want to visualize and interpret, so you will be doing we will be preparing the customized database based on different. So if you want to have a patient specific sequence or a patient specific files, so you will be doing a customized based on a single VCF file. But again, if you want to have a from a different databases, so you will have a from the multiple VCF files. So what you will get is that mRNA fast database, and it is the different parameters that you will be reading when you put in the use the tool, so to give different to get out different outputs according to your. So if you go and search, so here I'm just pasting the links in the chat box. So Cartadapt is also been ready. After looking at custom Proteogenomics one, so we will look at the Cartadapt data and then continue. So here you can see that you can download the example. So variant annotation using based on single VCF file. So you will annotate the variant using ANOVA, and from here you will build your customized database. And if you have multiple VCF files, that also can be done. So this is the output files that will come. So customized protein database, and also it will give a table contains that detailed amino acid change information. So whenever there is a change in the amino acid from the actual initial database or initial variants, so you will get that each row it will mention you the variant, so that you can cross check. So if you get a peptide sequence, and then you can cross check with your table and can identify that which position the variant is there, and you can try to analyze and interpret from here. So this is the for the custom prot bj. So you will get the full result for it. So now moving on to the cut adapt data that has been done. So right, you can see that this is cut adapt what version is there. So what is the command line parameters that have been given, okay, on the which are the two files that have been added. So total read pairs processed number is given, pairs discarded as untrimmed. So okay, so you can see that these were processed, read one, this one, and these are the filtered. So since why this is showing is reason is that trim been done, so this has been trimmed. So I just wanted to show you that how it is you can perform it. And so here there are different colors. So green here means that it is job has been done and there has been no error. And you can see the length 125, so everything is mentioned here, so you can check everything. Here's a JSON file, too long read, too short read, so everything is it mentions here. So you can check on the different data as well as download those data. So you can here from here you can download different data, so all those data. So this is from the cut adapt tool. So now if we go and look for the one of the custom Protbj that database how it looks. So after you get, so as I told that you will get an mRNA FASTA file, means protein FASTA file after your translation into 3 frame or 6 frame. So you can find out that this it gives information like this with the number of the chromosome, from which chromosome it is being identified with the ensemble sequence ID and on the protein sequences. So after this, when you this FASTA file you need to upload in the different protein search databases with your raw file, and then you can identify your novel peptides or novel sequence. So after using custom .bj, you will get an output in a FASTA file in this format with the sequence identifier and the sequences, from where you can try to get an idea and identify those sequences. So this was about that how you can use the Galaxy Web Server as discussed in the lecture, and how you can build up a customized database for your proteogenomics analysis. So now moving on. So there was this is the approach. So after you identify your novel discovery phase proteomics data, either through this customized database or either through the human UniProt database. So now after that you will need to identify those proteins. So now the one of those approaches that is becoming very much useful. So before going on, I just ask one. So if you have any doubt or anything, or can you use this software or not? So if you have any doubt, you can write in the chat or ask. So if not, so you can write that okay, means till that then we'll go to look at the skyline thing. So do you have any doubt or can I move? Okay, so let us okay, let us move here. So the different proteomics data needs to be gets validated. So the major validation things are generally dependent on antibodies, like for the ELISA or Western blot, but it went so after the targeted proteomics has become popular to validate those proteins that you have identified. So be it your mutant or the variant peptides or variant proteins, or and he discovered if has proteins for two different conditions. So you can go and target using different methods. So for this you need the skyline to build up your methods for the pair of transitions for a particular peptide, and also to analyze your data after this run. So and go to it. So you can go to the page, this skyline, the link has been pasted in the chat, and you can go and download it. Also you can check on the video of the skyline tutorial, and you can go and download the tool. So in this, generally, if you what you can check out is that after your data has been analyzed, so you will need so you will get these proteins with the peptides, and you can check on the different transitions. You can check on the different proteins, and then you will check on the different transitions and how it is matching with your library of the spectral library. And if you can see that it is matching with your spectral library, the proteins that you are looking for or monitoring those specific peptides and those proteins that you had found out in your data, and try to validate those. So once you can see that there's number of peptides having a number of transitions and matching with your spectral library from your shotgun data, or so then you can conclude that okay, so this proteins is been present, and also you can check on its different intensity levels for a particular peptide. So this is an example of how the data looks after being run in a triple quad mass spec, and how you can look. So here all these are called one transitions for the different pairs, for different q1 q3 these pairs for each particular amino acid, and you can validate those peptides and proteins. So now if you want to build that, how you want to build the transition list. So you can go and add different libraries from your data with some modifications. As you might remember that using uh, mascot, we need to have the structural modifications like carboamidamethylation as a, to keep it as a fixed modification. The digestion using trypsin and with maximum least keyword 0, and background human proteome as human cis proteome. And you can enforce the peptide uniqueness. So those proteins, if for the proteins you will be inserting those proteins having unique those peptides that will be unique to that particular protein will only be kept, and others will be removed. So other peptides will not be kept for building up your list. And then okay, so then you can insert your FASTA sequence, or you can insert your protein sequences. So if you can So you can see that human albumin you can insert, or you can insert ferritin, heavy chain, or any specific like globulins, immunoglobulins. So you can check for errors and then you can insert. So you can see that the peptides that are unique to this particular protein has been inserted, and it is the library that should look like. So now after that, after you prepare your samples, like the protein sample after digestion, and when you will prepare your samples and put in the mass spec, so what result you will get, so your data should match with this fragmentation pattern, with this spectral library, and if it matches with your particular spectral library, then you will get a good peaks, and you can say that the peptide belongs to that the you are monitoring, and you can conclude that yes, you can validate those peptides and the proteins. So in this way you can prepare. So now after this you will need to have those transition settings. So here you will give the filtered charge, so export, you can export the transition list. So you will give accordingly the instrument you want to check. So if you go, so if you open this Excel file, so you can check that for this particular peptide it will check out the RT window polarity, the precursor and the product. So you can see that the precursor will remain same for this particular compound, but with different m by z and the different collision energy. So as you had seen in the lecture how it has been done, so from in this skyline you can use this method to generate this list of those peptides that you want to monitor with all the collision energy, product ions and the precursor ions, and can give the instrument the instructions to look for this particular precursor ions at this particular to allow the peptides of these m by z ions only to go and to be filtered out, and the m by z for the product ions at the Q3 level, and finally identify the that these peaks and this precursor and product ion, that is transitions, are present or not in your sample. So this is how you can build up, and also it will give out different collision energies that will require to fragment your peptides in an optimum level. So in this way you can search it, and for the result file you can import the results. After this you will get the different uh, raw FASTA files, and then you can look at the results like what I showed in the PPT, where you can see that the different files for the different number of amount of samples, as many as number of samples you would want to run. So with all those samples run will give you particular uh, peaks, particular samples will have particular peaks for that particular peptides and the proteins. So here you need to align your peptides at a particular detention time, and then would like to check the intensity levels for those peptides and how much it is behaving differently. Is it uh, following the same hypothesis that you have derived for, or is it deviating from your hypothesis, or what are the how the your peptides or proteins are there in the sample. So once if the peptides are relatively stable, or you identify those peptides that are quite stable in nature with the different optimization in different pools, then you will automatically go and identify uh, I can identify and quantify those particular proteins at your level of active for your sample interest. So another thing is that for the mutated, if you identify a variant peptides using your variant database, so after you generate your variant database and with proteomics data, if you get a mutated peptide, then you can insert your then with the protein file you cannot insert the proteins that are already available or present in a non-mutated state, then you will analysis will go wrong. Then you need to insert the FASTA, and here you need to give with the FASTA format you need to give your sequence, particular sequence that has come as mutated. So after you got optional protein description, so you can give, then you can need to insert it. So you need to have the sequence that you will get. So in this way you can write the sequence for this particular mutation peptide, and then you can also you can build up a library proteome using the customized database that you have generated. So for example, if you in the peptide settings, so here background proteome is our human Swiss proteome that is present with all the normal proteins and the peptide sequences. So if you have built your own customized database with the protein sequences, so you can give the background proteome as that protein sequence. So when you give that as a background proteome, then when you give that as a background proteome, then what happens is that whichever mutated sequence comes will come from that proteome database, and when you provide that sequence for the faster sequence for that protein database, then you will not get any error, and also you will could add those peptides in your skyline list, and then you can build the transition list for those particular peptide which you have seen that it is coming as variant. And once it is coming as variant, then you can go ahead and run in your sample to validate that yes, that mutant peptide that has come from the variant database is showing the same in the targeted approach when you are providing a particular set of M by Z for those particular peptide, and it is showing the same and it is and the library and your data is matching, and then you can validate your peptide or the protein findings, and you can find out the significance level of the variants as well as correlate those with your disease and can analyze those significantly. So this is how you can change when you find, so you can change your background proteome as well as you can change your you can insert the particular sequence which you want to monitor as, which have come as a modifiers. So for example, in the Galaxy Web Server, in the lecture we have seen that with your MGF file you can write it in the, you can with your sequencing data you can directly match and you can find out the different sequences and which are the modifiers at that particular amino acid level. So with all those sequences you can keep and you can insert it at once in a skyline and can prepare the transition list from where you can identify those particular uh, transitions for that mutated peptides which have a change in M by Z level, which will have a particular change because of the change in the amino acid level. It will have a change in the M by Z level of that particular peptide. So from here you can try to identify. So this is the overall workflow. So if you go ahead and look at this paper and go and different tools, so you will get an idea of the different tools that are available, and with every tool their pros and cons, and to use which tool you want to use depending on your particular application. And there are a huge number of tools that are integrated in the Galaxy Web Server, and you can download your own data or you can use your own data and use Galaxy Web Server for uh, analyzing your data. And finally you can also, with your peptide sequences, if you identify some peptide sequences, you can validate those using Skyline, using first prepare the transition list of all those peptides or the variants that you have identified through your proteogenomics analysis using Skyline, and then can validate those, and with the background proteome of the customized database that you have generated using custom prot.bj by annotating, by aligning by the genomic sequences that you have got. So in this way you can have identify those variants, assess their impactful variants, and also you can validate those in the sample. So if you have some data from the genomic data, if you download and try to find out the variant database, and with if you have the protein sample or the protein lysate, so you can find out that though there will be not fully matched, but still you can try to find out some variant peptides are there or not. So this is, so this was all for the week 6. So if are there any questions regarding it? So I have provided the links for the custom prot.bj as well as for the paper, as well as for the Galaxy, as well as for the Skyline tutorial. So if there are any question for the previous week or this week, or any doubt regarding, so fully not, so everything is clear. So this was for the week 6 live session for the course Introduction to Proteogenomics. So yes, so thank you for joining. So okay, so I'll end the session today here. So thank you.